Of skeletal muscle. We're going to go over just basically the membrane physiology as well as skeletal muscle physiology. Types of muscle, don't need that. Do you want me to go over that or no? Just the types? No. Let's just get back into it. Wherever he was. All right, so Titan, that's one that he talked about. It's basically like a spring protein that's keeping everything aligned properly so that the myosin heads and the actin filaments don't get into places where they shouldn't be. It also, right here, it says molecular spring responsible for passive elasticity of the sarcomere. This is part of the reason why when you stretch a muscle out, it goes right back into the same position, is from the actual springiness of Titan. So our thick filaments are myosin filaments, thin is actin, and Titan is actually not shown. Inside the space between two Z discs is a sarcomere. The Z disc is where the actin filaments are going to be connecting. And the A band is where the actin filaments and the myosin tails are going to be overlapping. So I like to think of A for actin. The I band is going to be this space right here where the actin filaments are only. So we are only seeing the thin actin filaments. That's going to be your I band on either side of the Z disc. And then the myosin will pick up in that A band. And the H zone is where just myosin is present. So right here is just myosin, and that myosin is anchored to those Titan protein coils. And the M line is just in the middle of the myosin, so where the myosin extends from either side. So I like to think M for middle and M for myosin. So M, middle, myosin. And there's our Titan filaments that are coming in to bind those myosins. Here is our relaxed sarcomere, where everything is spread out, and our contracted sarcomere. Let's get more into the chemical action of it. Okay, so we're getting into the sliding filament theory. We're already assuming that we've come from a neuron. So actually, let's step forward to our neuron and talk about what's happening here. So here's our neuron. The action potential is going to be traveling down the neuron. We already talked about membrane potentials. So when you have a voltage-gated sodium channels open, the sodium rushes in, and that's going to depolarize the membrane. That action potential travels all the way down. Now when we get down here, these are our little synaptic vesicles. The synaptic vesicles are in the synaptic terminal, and they're holding our neurotransmitters. In this case, because we're going to skeletal muscle, they're going to be holding acetylcholine, because we know that from DAA that our neurons going to skeletal muscle are nicotinic, and they're releasing acetylcholine. So what happens here is this depolarization travels down the membrane, we get to the synaptic terminal, and we need to get this synaptic vesicle to fuse with the membrane down here so we can spit out our acetylcholine. So the voltage-gated calcium channels are going to open. So that action potential runs down. It triggers voltage-gated calcium channels, and that's important because calcium is what's going to signal our vesicle to merge with the end. So our calcium comes in after the action potential depolarizes the very end of the axon. Calcium comes in, triggers the vesicle to merge with the actual plasma membrane at the end of the synaptic terminal, and that spits acetylcholine out into the synapse. Now that acetylcholine is going to need to find a home, and that's not going to be a voltage channel on the actual motor end plate. So the motor end plate is what we call where the neuron is interacting with skeletal muscle. So these are going to be ligand channels, which means basically that you have a receptor that is being stimulated by a chemical rather than by an electrical gradient. So our ligand acetylcholine channels are, our ligand stimulated are stimulated by acetylcholine, and that's going to open up our sodium channels. So they're ligand stimulated sodium channels. And the sodium is what we're using to depolarize the membrane again. And it's important to keep those two separate. Is when we're talking about neuron to neuron communication, we're just talking about the conduction of the signal, so you're going to have all the things are the same. But when we get down in here into the motor end plate, now this is a muscle cell. So keeping that separate in our mind, we're going to depolarize the membrane of the muscle cell the same way, but we're going to want different end products. So here is another picture of that. So after these acetylcholines bind to our ligand-gated sodium receptors, sodium channels, then the sodium channels are going to open, sodium is going to flood in, and it's going to flood in, and when sodium comes in, it depolarizes the membrane of the muscle cell itself. And that depolarization is going to go through the sarcolemma, which is the wrapping on the outside of our sarcomeres and of our muscle fibers there. And it's going to spread through the T-tubules, so the T-tubules perforate all through the muscle and spread that depolarization out. And eventually what that's going to do is it's going to reach that protein he was talking about here at the end, which is our DHP receptor. So it comes down to our DHP receptor, it's going to activate that, which will pull on this string. And then this, I think he said, was RYR receptor, and I can't remember what the particular words are. I don't even think he has it on the PowerPoint. But that will open this up. This is our sarcoplasmic reticulum, and then the calcium is going to come out and flood into the sarcomere. So how are we getting the calcium out? We're using sodium to depolarize the membrane, which triggers our voltage-gated DHP receptor. That's going to pull and open this receptor here, the RYR receptor, which allows calcium to flood out into the sarcomere. So our goal is to get calcium in here so that we can have muscle contraction. So let's go back now to this cycle. Where did it go? Just further back. There it is. So this cycle basically picks up with the assumption that now calcium has already spilled out. So we've already had our neuron, everything with the sodium and the depolarization of the membrane has happened. That sodium rushed in, that voltage changed, and now our calcium has come out of the sarcoplasmic reticulum and into our sarcomere, and it's now diffusing out through the space. So we've got a couple of things going on here. The first one is that we have our, where's our actin? That's myosin. Well, our actin filament is going to be up here in blue, and then here there's a strip and a little ball on the strip. And so the actin filament is all of these little blue chains here, and it's going to have binding sites for myosin, so myosin can eventually attach. But those binding sites are covered, which is preventing myosin from attaching. And so what's going to happen now is that calcium comes in, and on this chain, which chain we would call the TTC, which is troponin tropomyosin complex, well, there's a little ball on that chain, and that is the actual troponin part of it. So you have troponin tropomyosin complex. So troponin is actually going to be able to bind with calcium. So that calcium comes in, and our calcium is going to grab that troponin. That causes a conformational change. So it's going to change its shape, and that pulls this whole strand away from those binding sites. So now the binding sites on the actin filament, which is our thin filament, are exposed. And when those binding sites are exposed, that will then allow myosin to connect to it. But that doesn't happen right away. What we're going to need is we're going to need ATP. So you can see up here, if we were starting with our myosin attached in step 1, right? Here's our myosin head, and it is bound to that actin, and it's got an ADP and a phosphate on it. So it attaches. The next thing that's going to happen is that ADP and that inorganic phosphate are going to detach during the power stroke. So during the actual pulling of the myosin head, that conformational, I can't remember if he said it was conformational change, but it pulls. That's when our ADP and our phosphate are going to detach. And that's important because that's what leaves the myosin head stuck in that position. So this is the power stroke. This is where you're actually going to get, when all of these myosin heads move, they're pulling the Z-disc on either side closer together, and that's our muscle contraction. So what's going to basically unhook myosin once it's stuck is another ATP molecule is going to come on. So we're out there in our skeletal muscle, we've got our mitochondria that are just generating energy for us. Now, if we're not under high demand, then we're doing aerobic respiration, so we're using the TCA cycle, we're getting lots of ATP out. So it's no problem to meet our energy needs. If we are under high demand, then we're using anaerobic respiration. We're getting less ATP out, and that's where you can actually get into muscle fatigue and whatnot, where the myosin heads can't detach, and you would call that like a cramp or a spasm of the muscle. But so in a normal case, another ATP comes along, it binds to that myosin head, and the act of it binding is going to snap that myosin back down to the relaxed position. So you can see here, we're in this more relaxed position. Now, if this process needs to repeat itself, like we need to keep contracting the muscle, then that will just repeat. That ATP, the myosin head has ATPase activity, enzymatic activity on it, which will pop that ATP off into an ADP, and it'll lose phosphate again, and then that myosin head will reattach. So if you need the muscle to contract again. But let's say we don't want the muscle to contract anymore, so that doesn't happen, and the calcium at this time that was dealing with actin is now coming back into the equation, so we're thinking about what calcium is doing again. So I asked him after class, and he said that after the calcium rushes in and binds to troponin and exposes the binding sites on actin, then the calcium pumps are slowly trying to pump calcium out of the cell and back into the sarcoplasmic reticulum. And so in the area around the particular sarcomere, there's less calcium concentration as calcium is getting pumped out. And so that creates a gradient where the calcium wants to go to the low gradient around it. So that will cause the calcium to unbind from troponin, is following that concentration gradient. And once it's unbound from troponin, troponin and TTC goes back onto actin, and those binding sites are now covered. So myosin, even if it was trying to, can't cause muscle contraction. That calcium gets pumped either outside the cell completely, so extracellular, where it can be used to start that process over again, or it gets pumped back into the sarcoplasmic reticulum, which is basically to be used for the next time that we get an impulse from that neuron that depolarizes the membrane. So that's basically the process from start to finish. It's important to we kind of bounced around in order, but so you have the depolarization within the neuron that spreads down. That's going to trigger our voltage-gated calcium channels. Calcium is going to come in and stimulate our vesicles to migrate to the plasma membrane. Once they're at the plasma membrane, acetylcholine comes out into the synapse. Acetylcholine is going to bind to our ligand-gated sodium channels, which open and sodium rushes in. That sodium is going to depolarize our muscle cells, and once that depolarization has occurred within the muscle cell, I don't know where that slide is. There it is. So once that depolarization has occurred along the sarcolemma and all through the T tubules, that's going to stimulate this DHP receptor, which is voltage gated, because remember we're dealing with depolarization, so this is a charge gated protein. So the DHP receptor is stimulated, it pulls, it lets calcium out of the sarcoplasmic reticulum, and that calcium floods the sarcomere. We go back to our sarcomere, and that's where that whole process starts. So calcium comes in, it binds to troponin, that pulls TTC away from the binding sites on the actin molecule. Then at the same time, our myosin head is going to cleave an ATP, creating ADP and in a free phosphate. That causes it to come up and snag the actin filament, pull on it, Z discs get close together, that's muscle contraction. The ADP and phosphate are actually what's leaving when that muscle contraction, the power stroke, occurs. Then it's just hanging out and stuck until another ATP comes along. The ATP comes along and binds to the myosin. That's going to cause another change. Myosin comes back into the relaxed state. At this point, calcium should be diffusing out of the sarcomere. The calcium pumps are pumping it out, either back to the sarcoplasmic reticulum or extracellularly outside the cell. And then TTC goes back over the binding sites, making them inactive, and you have resting state in your muscle. And that's kind of the whole thing from nervous impulse all the way through muscle contraction back to resting state.